My name is Matthew Hall. I'm a group leader of biology at the National Centre for Advancing Translational Sciences, which we lovingly refer to as NCATS. It's a real pleasure to be here today. I suppose it's a real pleasure to be here at all. I'm very thankful that WALS wasn't shut down. And it's a special pleasure to be introducing Dr. William Kaelin today. Bill's a professor in the Department of Medicine at Dana-Farber Cancer Institute and at the Brigham and Women's Hospital as part of Harvard Medical School. And he's also been a Howard Hughes Medical Institute investigator for 20 years now. It's a highly distinguished career, began with undergraduate and medical degrees at Duke University, and he then completed a residency at Johns Hopkins University. And while he was there, he worked in David Livingston's lab studying tumor suppressor proteins before moving up to Boston. He's served on a ridiculous number of advisory councils and boards, and I'll just mention a couple. He served as a member of the NCI Board of Scientific Advisors and also the AACR Board of Trustees, and been elected to the National Academy of Sciences as well. His work has won him numerous awards, including the Albert Lasker Award for Basic Medical Research 2 years ago. His career is focused on understanding the role of mutations in tumor suppressor genes in cancer and oxygen sensing as well, which has really provided a key understanding of the role of proteins such as retinoblastoma, von Hippel-Lindau, its role in hypoxia and P53 tumor suppressor genes. In recent time, he's also done some really critical work in helping us to understand the role of mutant IDH1 in oncogenesis and cancer progression as well. But Professor Kaelin's also served on scientific advisory boards for a large number of pharmaceutical and biotech companies. Currently, he serves on the board of Eli Lilly as well. And it's really this privileged duality of insight, I think, into the molecular basis of cancer and also the human exercise and challenge of therapeutic development that has really driven some of the insight that he's going to provide you in today's lecture. And something we've talked about a lot at NIH as well, rigor and reproducibility in scientific research, which is absolutely critical. So please join me in welcoming Professor Kaelin to speak today on preclinical cancer target validation. How not to be wrong.
Okay, is there a trick to getting my first? There we go. Thank you very much, Matt. It's a pleasure to be here. I think the genesis of this invitation was Matt heard me give a variation of this talk at Cold Spring Harbor last summer, and he said it would be great if you would come and give this talk, and you're supposed to dance with the one who brought you, so I'm going to comply with that request and give a version of this talk. But this is really a talk that sort of had been swirling around in my head for a number of years now. First of all, you might have noticed the peculiar title to my talk, which is partly based on this book, which I highly recommend, especially to the young people. So this is a marvelous book about the power of clear and logical thinking in day-to-day life, and he gives a number of wonderful little examples and vignettes, some from things that are happening currently, some from things that happened historically, where the knee-jerk obvious answer turned out to be conspicuously wrong. But if you thought about it a little more deeply, you got to the right answer. I hope some of our friends in Washington will also read this book. So anyway, I think if you read this book, I think you can see how logic colors everything we do. And now I'm going to try to apply some of these principles to what we do in the laboratory. And as I said, part of the motivation for this talk is I had come to know from my interactions with our friends in pharma and biotech how often they were having difficulty reproducing the results that we were publishing in our favorite journals. And you might be familiar with this paper or the following paper where two pharmaceutical companies, I think it was Amgen and Bayer, said that on the order of 50 to 80% of the target validation studies they were attempting to replicate were either non-reproducible or were not sufficiently robust to allow them to proceed with a drug discovery effort. And frankly, this problem, I think, as you know, has gotten increasing attention. And when I joined the Lilly board, I went to the head of research at Lilly, and I asked him about this 50 to 80% non-reproducibility problem. And he turned to me and he said, in our hands, it's 90%. And I waited to see if he was going to start laughing. And then I realized he was dead-faced serious, so then I knew we had a problem. And you don't want to be discussing this at the Thanksgiving table with your non-scientific relatives, like what in the world are you doing with these precious resources that are being entrusted to you? Why is everyone talking about the reproducibility problem and the robustness problem? Now, the two papers I just alluded to, admittedly, were cancer target validation papers, but I want to highlight this is not unique to cancer. This cuts across all therapeutic areas. And although the examples I'm going to be talking about today in terms of things you might think about in the lab are really driven from our own work in cancer, I think you'll hopefully see that some of the principles apply more broadly. Now, the first time I gave this talk in Cold Spring Harbor, someone jumped up as soon as I finished and said, that's not fair. A lot of the problem is really robustness. If you really look under the hood, a lot of it is really more robustness than literal reproducibility, and that's true, that in some people's hands, the major problem is robustness. But then you have to at least agree on what does that actually mean. So when I ask my trainees, what does robust mean? They think it means great, big, wonderful. But of course, that's not what robust means. Robust means the ability to withstand perturbations. So for example, on my plane down, there was a co-pilot. That's partly to increase the robustness of commercial air travel. If a server goes down someplace, the internet doesn't suddenly crash. The internet, of course, is highly robust. But in our world, this means if the result is true, but it's only true at pH 7.30, but it's not true at 728 or 732, already you might be concerned about robustness. If it's true, but it's only true in your favorite cell line, and it had to be Friday, and the Red Sox had to have won the night before, it might be true, but that's not robust. So we need things that are highly robust. Now, how did we get to this problem in terms of reproducibility and robustness? And I'm going to argue part of the problem relates to this slide. When I was a postdoc in the late 80s and the early 90s, you were a second-class citizen if you did translational research. And then in one generation, the pendulum swung all the way in the other direction, such that if you don't do translational research, you're suddenly a second-class citizen. And so I think what that's done is it's had the expected outcome that increasingly people feel like no matter what they're working on and no matter what they've discovered, their last figure better look something like this. So I can hear the clock ticking as I'm reading papers. Figure one, I'm really interested, but then I can see they're going to race to get to this figure, which often includes a Kaplan-Meier curve where expression of the investigator's favorite gene is linked to prognosis. And on the right, they've knocked down the gene in some mouse xenograft experiment, and there's been some measurable effect on tumor growth. And now they've achieved impact and clinical relevance, and so they can justify what they've done. So, of course, I think there are lots of problems here, and we're going to talk about them in greater length. But before we deal with these problems, I think we have to step back a minute and just go back to teaching our trainees some basics in terms of how to think logically and clearly, and to use words that actually have precise meanings, because many times in biology, of course, we're encountering correlations, and then the question is, are they really causal? And so, for example, I like to review with students that two things can correlate with one another, say A and B, because maybe A causes B, maybe B causes A. In biology and medicine, there's frequently a hidden confounder that's causing A and B, but A and B don't cause one another. And then in the era of systems biology and multiple hypothesis testing, it's pretty easy to find things that simply correlate by chance, but there's no causal relationship. I also encourage students to try to use words that have precise meanings, and so try to avoid wishy-washy words like is associated with or linked to, but to use words that maybe actually tell me what you think. And I find that two words that are very useful are the words necessity and sufficiency, and I'm always surprised at how many students are not familiar with these concepts. But of course, A is necessary for B means if you don't have A, you won't have B, whereas A is sufficient for B means if you have A, then you must have B. Now, there may be times when I say what I just said a moment ago, which is, we need to teach students this, but I can tell you occasionally I'm reminded that it's not just students. And so, for example, when BRAF mutations were first identified in malignant melanoma, I went to more than one SAB meeting where a tenure-track professor-type person would say, well, BRAF mutations won't be good targets in melanoma because you can already detect the mutations in the benign nevi that we think are, in some cases, the precursors to melanoma. And I would just scratch my head because, of course, the fact that the mutations are already detectable in benign nevi tells you the mutations are not sufficient to cause melanoma, but that doesn't address the therapeutic question of whether those mutations are necessary, at least in the melanomas that are formed at the hands of a BRAF mutation. And of course, the answer was yes. In a fully formed BRAF mutant melanoma, there's an ongoing requirement for BRAF. Another more subtle one is the sort of implicit sort of suggestion, or the thing people would tell you about their darkest fears if you gave them truth serum. And that is, many people, if you gave them truth serum, would throw up their hands and say, I'm not really sure we can do this. Cancer is just too complicated. There are too many mutations. There are too many drivers. We will never get there. But of course, these people are also conflating necessity and sufficiency, because the fact that there are multiple mutations in many common tumors, multiple drivers, tells you that perhaps multiple mutations were necessary to cause that cancer. But in a perfect world, then it would be sufficient to undo any one of those mutations. So think of a combination lock, for example. So if I told you that 3 tumblers had to be in position for the lock to open, you would say that's good because I only have to undo one of those tumblers to keep it from opening. Now, I don't mean to suggest we're not going to need combination chemotherapy in cancer. There are lots of reasons why we're going to need combination chemotherapy in cancer, but it's not because N drivers equals logically the necessity for N targeted agents. Okay, and so that's just what I just said a moment ago. So now let's return to the Kaplan-Meier curves and the xenograft experiments. So let's start on the left. I sometimes call this the marriage of the gratuitous and the contrived, because you know full well, first of all, that what the person has done is they've searched as many databases as possible until they found one where the expression of their gene was linked to a bad outcome. And furthermore, they're trying to seduce you into thinking that not only is this correlative, this is causative. You better study my favorite gene. My favorite gene is really important because, look, it's associated with a bad prognosis. And they're doing a sleight of hand here frequently, which is a shortcut we've all taken on occasion, some more than others, which is you have a story where you have a correlation. You can imagine biological plausibility, such as expression of my favorite gene might be bad for the following reasons. And so you simply declare a victory and say, okay, that's good enough for me, that's causation. So the danger, of course, can be illustrated several different ways. So here's a theoretical Kaplan-Meier curve for patients with chronic lung disease who either have or have not ever been placed on a ventilator for their chronic lung disease. I can guarantee you the curves would look something like this. Any doctor wouldn't be surprised that if you have chronic lung disease that's bad enough that at least on one occasion you went on a ventilator, you're probably going to do worse than the rest of the patients. But I hope none of the people would therefore say, well, we better get rid of these ventilators. And these ventilators are causing these bad outcomes here. So you laugh at that one, but I can tell you in our field there are much more subtle, much more insidious examples of this. So for example, I work on the hypoxia-inducible transcription factor HIF. I can tell you there are 100, if not 1,000 examples of figures that look like this, where hypoxia within a tumor has been associated with a bad prognosis, upregulation of HIF has been associated with a bad prognosis, or expression of some HIF target gene has been associated with a bad prognosis. And of course, the seduction here, which might be true, is that maybe hypoxia and HIF causes the bad prognosis, the bad behavior of the tumor. But I could also imagine, and there's some support for this now, that there might be some tumors where hypoxia causes bad behavior of the tumor, but HIF is simply a marker for hypoxia. And I can tell you my favorite explanation for these data are that aggressive tumors outgrow their blood supplies, become hypoxic, and upregulate HIF. So now the heart, you flip the cart and the horse. So I think you have to be very careful with inferring causality from these kinds of curves. And at a minimum, this suggests that being associated with a bad prognosis is not sufficient to declare that this is likely to be a good target. But is it necessary? How about the opposite? How about if your target's associated with a good prognosis? Should that disqualify it as a target? And once again, I heard tenure-track professor caliber people, when IDH mutations were first identified in glioma, argue that we should not target the IDH mutations in glioma because, at least at the time, it appeared that IDH mutant gliomas had a better prognosis than IDH1 wild-type gliomas. In fact, one person was even on the stump speech saying that Agios, and I have my financial conflict of interest, was guilty of medical malpractice if they even imagined treating patients with IDH mutant gliomas with a drug that would inhibit the mutant IDH. Now, of course, if you follow this sort of Monty Python-esque logic, you would conclude that we should never have developed tamoxifen for the treatment of breast cancer, because it is true that amongst breast cancers, the ER-positive breast cancers have a much better prognosis than, for example, triple-negative. But that doesn't tell you that in the ER-positive breast cancer, there isn't an ongoing need for the estrogen receptor. It just tells you that the biology of those two tumor types is completely different. And so I would conclude from this part of the talk that being associated with a poor prognosis is neither necessary nor sufficient to make you a good cancer target. So I think we should maybe de-emphasize a bit the gratuitous Kaplan-Meier curves in Figure 6, where we're trying to seduce you into thinking this is adding further credentials to my target. So now let's leave the gratuitous Kaplan-Meier curve and now go to what I call the contrived tumor xenograft experiment. So the first thing that you should note about these experiments, if you do sort of a back-of-the-envelope calculation, so what's been done, for those of you who don't do tumor xenograft experiments, is you typically inject on the order of 10 to the 6 cells frequently into the subcutaneous space, and then you typically terminate the experiment when the tumor is about a cubic centimeter. And if you do the math in terms of the doublings that have happened here, this publishable difference could be as trivial as going from 10 doublings to 9 doublings over the course of, say, 2 or 3 months. And what's particularly problematic about the well-used, well-worn xenograft assay and its equivalents is that a screener would tell you this is a down assay. You took some complicated system and you made it work slightly less well, such as by knocking down the expression of your favorite gene. Now, people who do chemical and genetic screens hate down assays, and they would much rather have an up assay for several simple reasons. First of all, in this fictitious screen where here are my signals and here are my samples, even before I got my laser pointer out, you probably saw the up in this screen, and usually in an up assay you have a better signal to noise. In fact, if the screen is noisy enough, you can't see the downs, but here are some of the downs down in the weeds. But for us, the more important problem is there are more uninteresting ways to make a complex system work less well than there are to improve it. And many of these, from a drug discovery point of view, would be kind of trivial to us. So, for example, when a professional athlete gets injured, their performance almost always gets worse. If the students want to do an experiment, they can go out to their internal combustion engine and start removing parts and come back and tell me how often the car goes faster. The more things that are necessary for the system to operate properly, the more ways there are to make it work less well. And again, from a therapeutic point of view, some of the targets that would be revealed in a down screen will be relatively trivial or uninteresting. And so a screener would say, get away from down assays and try to do up assays. And so just to show you this in kind of quasi-real life, back in the day when Rick Klausner was still at the NCI, his group and our group were working on the VHL tumor suppressor protein, the VHL gene having been isolated by Marston Linehan and others. And we and others showed that the VHL protein is part of a ubiquitin ligase that in the presence of oxygen targets the alpha subunit of the HIF transcription factor for degradation, whereas when oxygen levels are low or the VHL protein's been mutated, now HIF alpha can accumulate and dimerize with its partner protein ARNT and activate HIF target gene. So this is the current, or was the current, and it still is the current view of how the VHL tumor suppressor works. Okay, so then a question is, well, is HIF, and in particular deregulated HIF, driving the formation of these tumors? Or is it simply a marker for the integrity of the VHL protein? And again, you can start to spin a story based on plausibility that HIF is actually causing the cancer. But to be honest, 10 or 15 years ago, we thought maybe it's just a marker. And so Keiichi Kondo in the lab, back when this was sort of a new technology, it's not a new technology anymore, created a short hairpin RNA that would downregulate HIF2 alpha in a VHL defective kidney cancer cell line compared to an empty vector. He implanted the cells into a nude mouse, and as expected, the empty vector cells still formed a tumor. When he knocked down HIF2 alpha, and I'm not showing you the confirmatory Western blot, now these cells could no longer form a tumor in nude mice, so that's sort of the hoped-for, expected result, that maybe HIF2 alpha really is the driver here. When you knock down HIF2 alpha, you see diminished tumor growth. But again, if you're really being honest, this is a down assay. Okay, what was happening before is not happening now. And again, whenever you do a down assay, you're really setting yourself up for what's called the off-target problem. So you have some perturbant. It could be a drug. It could be a short hairpin RNA. It could now be CRISPR. You modulate the target, and you get some phenotype, in our case, decreased tumor growth. But if you're really being honest with yourself, especially in a down assay, maybe this hairpin that we designed does 10 things, and it's one of those other things that's really responsible for the decreased tumor growth. It's not the knockdown of HIF2 alpha. And so you really have to start to think about how do you control for these things. And just to show you how we're drowning in down assays, I think many of you would have appreciated that the xenograft assay is a down assay. But look at the way we do a lot of our pharmacodynamic studies. So based on our interest in HIF2 alpha, when Wagao in the lab had done a screen for chemicals that would downregulate HIF2 alpha, even in a cell that lacked the VHL ubiquitin ligase. That would be kind of interesting if the problem here is really the loss of VHL activity. And he did a screen, and he identified chemicals that, sure enough, downregulate HIF2 alpha, as shown in this Western blot, a nice dose titration. And this is a publishable result. I see papers all the time on my favorite HIF inhibitor. But if you're really being honest, and I always tell the students, the least interesting explanation for your data is the explanation for your data until you prove otherwise. So the rosiest interpretation is, wow, this is some new direct degrader for HIF2 alpha. But there's a sleight of hand here. And so once you realize this is a down assay, your radar should go on for, am I getting misled here? Okay, it's a down assay. So the sleight of hand that no one ever tells you is almost invariably the person's favorite protein has a shorter half-life than the load and control. So even in cells lacking VHL, HIF2 alpha is still turning over pretty rapidly. So I could do this with cyclohexamide. I could do this with actinomycin D. If I carefully titrated the dose, I could do this with Clorox bleach. But that's not what we're looking for here. We're looking for something that's going to be more specific. And I won't tell you in the interest of time that he's gone on to do other assays that say that at least this is specific for HIF2 alpha, and for example, it doesn't affect the paralog HIF1 alpha. So now I've told you the ingredients for how I think we got to that 50 to 80% problem with reproducibility and robustness. A typical cancer biology experiment today, or cancer pharmacology experiment, you start with a perturbant, which parenthetically is usually a loss-of-function perturbant, but some of the same considerations would still apply for gain-of-function perturbants. We then do any of a number of down assays, decreased proliferation, decreased viability, decreased tumor growth. Then to really complicate things further, we not infrequently do multiple hypothesis testing, such as when we do high-throughput screens. And then if you really wanted to make this a disaster, you would set up a promotion and publication system where you reward people for the positive results and the negative results get buried. So how do we walk back from this precipice? So the good news is there actually are solutions for most of these problems, and I didn't have to invent any of them. But I think all of us have to maybe roll up our sleeves and start implementing them. So a geneticist would tell you one way to deal with this problem is a rescue experiment. And the rescue experiment would be to discover or identify or create a target that is functionally active but now is insensitive to the perturbant, and ask if you can reverse the phenotype. And so, for example, let's go back to the Noon mouse xenograft experiment. I'm proud to say that even back in the day, we appreciated that RNA interference might have off-target problems. I must say I underestimated, I think by an order of magnitude, how many off-target problems they have. But we were, I think, even then cautious enough, and Keiichi was cautious enough to realize he better establish this is on target. And so the classical way for an shRNA experiment of the one I just showed you was to now engineer a cDNA for HIF2 alpha that's going to encode a transcript that's no longer recognized by the short hairpin RNA because he's introduced translationally silent mutations into the mRNA. So this mRNA still encodes wild-type HIF2 alpha, but the transcript is insensitive to the hairpin. And then when he introduced this rescue construct, now he restored the ability of these cells to form tumors. And when we've done these sorts of gain-of-function and loss-of-function experiments, we actually came kicking and screaming to the surprise that at least in kidney cancer, and the rules might be very different in other cancers, the bad actor here is the less well-studied member of the family, HIF2 alpha. And if anything, HIF1 alpha seems to be acting as a tumor suppressor. And in fact, many kidney cancers silence HIF1 alpha altogether. So my dream 10 years ago was that maybe someone would develop a small molecule that would target HIF2 alpha. But many well-intentioned people told me that was impossible. But fortunately, Kevin Gardner and Rick Bruick at UT Southwestern did a screen in their academic labs, and identified chemical matter that could bind to a previously unappreciated pocket in HIF2 and prevent it from dimerizing with its partner protein, ARNT, and prevent it therefore from activating transcription. These were then out-licensed to Peloton to make them more drug-like, and this turns out to be a tool compound that I think is 2 atoms removed from the lead compound that went into the clinic, and they were kind enough to allow us to use this molecule. So Chin Cho in the lab did what any of you would have done with early access to this chemical if you had an interest in kidney cancer. So here's a cell line, a kidney cancer cell line that's VHL null. She's treating with 0.2, 2, or 20 micromolar of the drug, and she's going to measure these 6 HIF-responsive transcripts by real-time PCR. So the first thing she noticed is that the transcripts for HIF2 itself were unaffected. That's good because HIF2 doesn't autoregulate itself. The second thing is that of these other 5 are canonical HIF targets, and 4 of the 5 were downregulated by the drug, and the exception turned out to be quite valuable because BNIP3 is a HIF target, but it's only regulated by HIF1 and not HIF2. So by this and other measures, this drug is really having the intended effects on HIF and does appear to be relatively specific if you go on to do RNA-seq. Parenthetically, one other thing, you see a dose titration here. I can't get over the number of cancer pharmacology papers where you really have to do a lot of work to figure out what concentration is being used for the drug, because I can tell you at these two lower concentrations, this is an exquisitely specific HIF2 inhibitor. At 20 micromolar, all bets are off. This starts to become rat poison, okay? And so I think it's really important that we remember that. Okay, so now you do the obligatory xenograft experiment. We did it slightly fancier. So here we're doing a luciferase positive version of a renal carcinoma cell line that's been now injected orthotopically into nude mice. So these animals have unilateral or bilateral tumors treated with vehicle or the inhibitor, and then you follow using luciferase imaging quantified here, the animals that got the vehicle and the animals that got the HIF2 inhibitor. And lo and behold, as she had hoped, with the HIF2 inhibitor, you see stasis or, in fact, frank regressions. And then because we're often criticized, I think appropriately, for studying models of organ-confined cancer, Chin obtained from Joan Massaquet a metastatic version of the cell line that will form pulmonary metastasis-like lesions after tail vein injection. She did the same experiment but got the same result. If you treat the animals with the HIF2 inhibitor, she saw nice tumor control compared to the animals treated with vehicle. And now here's the Kaplan-Meier curve used in a purely descriptive sense. I'm not trying to make any causal inferences yet, but this is simply to describe the outcome of the mice treated with the vehicle or treated with the HIF2 inhibitor. And you want to go running up and down the hallway, high five, let's call the editor, let's write the paper, because this is what we had kind of wished and hoped for. But if you're really being honest, again, everything I showed you was based on a down assay. It was decreased transcription. I didn't show you, but decreased soft agar growth. It was decreased tumor growth in a kidney. It was decreased tumor growth in the lung. Maybe this drug really does inhibit HIF2 by pharmacodynamic criteria. It does. But maybe it also inhibits A, B, and C. And maybe A, B, and C is really responsible for the anti-tumor activity. So again, there's a playbook here. And the playbook is, can we come up with a target that's now been engineered or discovered to be resistant to HIF2 So here I would admit we got wildly lucky, because Gardner and Bruick had already identified a point mutation in HIF2 that would occlude this pocket in HIF2 and prevent the drug from binding. That's just shown over here. So then what Chin did was she took her renal carcinoma cells, and since these cells will tolerate loss of HIF2 under standard cell culture conditions, she simply CRISPR'd out the endogenous HIF2 and then replaced it with exogenous wild-type HIF2 or this putative drug-resistant version of HIF2 and shown here are the Western blots not only for HIF2 but for a HIF target such as NDRG1. But notice that for the putative drug-resistant version, if anything, it's slightly enfeebled as a HIF activator, but that's good, because for the students, if you're going to introduce a bias into your experiments, it would be better if the bias was working against your hypothesis and not in favor of your hypothesis. So now, for example, we can go back and do those RT-PCR assays looking at those HIF target genes, and you can see in blue the result I showed you a moment ago, whereas in red are the cells that now have the drug-resistant version of HIF2 and you largely reverse the effects of the drug on transcription, so that's good. You can also reverse the effects largely in those efficacy experiments I showed you earlier. So here's the pulmonary metastatic model. Here the animals have the drug-resistant HIF2-alpha, and you largely prevent the effect of the drug compared to the control cells. So by this and other criteria, we think, at least in these mouse models, this drug is acting on target. So then we committed a almost mortal sin, and the mortal sin was, in the paper we submitted, we said we've done this experiment with 8 different VHL null renal carcinoma cell lines, and much to our surprise, 3 or 4 were very sensitive, just like the review articles say they should have been, and 3 or 4 were, quite to our surprise, completely insensitive to the HIF2-alpha inhibitor. And when we went back and looked and we CRISPR'd out HIF2-alpha, they were still happy as a clam. And so this suggested that for the first time there might be heterogeneity with respect to the dependence on HIF2 in VHL-defective kidney cancers, despite all the review articles, frankly, some of which I wrote, saying that we should be thinking about HIF2-alpha. And as you might imagine, I got a lot of pushback from editors and reviewers saying, well, if you could only have told us why those other 3 or 4 cell lines didn't respond, then yes, this would be a Nature paper. But you don't really understand this heterogeneity, and so until you do, this is not really ready for publication. And I said, look, fortunately this was a relatively senior editor. I said, don't you understand that this is exactly the problem, that most people, because of this type of logic, would have cherry-picked their favorite models that did exactly what they were supposed to have done and buried the non-responders and left them for another day, and then someone comes along and doesn't see the expected result when they study one of those models. Now, I don't usually win those arguments. I won the argument this time, but frankly, I'm glad I had the argument, because guess what, and to no surprise to the clinicians, when this drug went into the clinic in patients with heavily treated kidney cancer, I think the median number of lines of therapy was 4. So this is a phase 1/2 trial of the Peloton HIF2-alpha inhibitor. These are so-called swimmer's plots, so each bar here is an individual patient. The dotted line for reference is 1 year on the time axis here. So the good news is that some patients have had very nice responses. There was 1 CR, there were I think 6 or 7 PRs, and good disease control in this group. But, to be honest, there's a whole other subgroup that the tumors are just waving at the drug and saying we don't care. And so I think it's really important to have models that actually, in a faithful and honest way, depict this kind of heterogeneity. And it's actually created an opportunity for us to begin to understand the resistance mechanisms. Now, at this point, you might be saying, okay, but Bill, you're pulling a fast one here. I mean, how often does someone deliver on a silver platter a drug-resistant version of the target of interest? That doesn't happen that frequently. What do I do if I'm not so lucky? And that's a fair criticism. But the good news is there are now some technologies that are quite powerful, even if Kevin Gardner and Rick Bruick don't do all the hard work for you. So one that we've really become sort of enamored by is a technique that was used by George Daly and then Charles Sawyer to study resistance to Gleevec, the drug that targets BCR-ABL. And Charles has subsequently used this for other targets. And the idea here is you have an expression vector for your favorite target. Okay? You then propagate the vector in error-prone E. coli, such as XL1 red that you can buy from Strategene. The E. coli do the work for you. They pepper the cDNA with point mutations. You introduce this randomly mutagenized pool into your sensitive cell line. You treat with the drug, and then you sequence the cDNA from the survivors. And to me, one definition of robust is when something works the very first time we try it, simply following the instructions in the published manuscripts. That doesn't often happen, but it must be robust if it works that way. So just to give you an example, Abhishek Chakraborty in my laboratory had also identified that in addition to HIF2-alpha, VHL defective kidney cancers are hyperdependent on EZH1, and this had been revealed through some CRISPR and shRNA screens. And then we obtained from Jay Bradner a tool compound called EZ05 that inhibits EZH1, inhibits EZH2, and God knows what else. And he could show that the VHL null cell lines were very sensitive to this compound, but the question is on target or off target? So he did the trick I just outlined for you on the previous slide, and he was able to identify drug-resistant versions of EZH1 that are insensitive to the drug, and those drug-resistant versions of EZH1 now reverse the sensitivity to this compound. So we think this is on target as well. And then if you really want to be completely agnostic about this, because in the prior examples you had to make a pretty damn good guess about what the target was, there's also a technique that's been developed by Tarun Kapoor and now further optimized by Deepak-Nishawan, where you either obtain a naturally occurring mismatch repair cell line, or using CRISPR, you make your favorite cell line mismatch repair deficient. And then after a certain number of passages, so again, that cell line has accumulated lots of point mutations. Now you treat the cell line with your drug, and then you isolate the survivors and do whole exome and transcriptome sequencing from cloned survivors. And this has been used repeatedly by those two investigators to identify targets for drugs where, frankly, in some cases, the target wasn't even known. Okay, so we've talked a lot about sort of the classical rescue, get a version of your target that's now going to be insensitive to the perturbant, whether it's a modified mRNA, a modified cDNA, modified protein. But sometimes you're unable to do that. But there is a second trick, and that is if you understand the biology of your target well enough, and you can restore the downstream effector functions of your target, and that rescues the phenotype, that would also give you greater confidence that you were on target. So, for example, we and others have been studying these very interesting IDH mutations that have been identified in leukemias and brain tumors and other tumors. You may know that these mutants share in common that they lead to the production of millimolar amounts of this oncometabolite called R2HG. And the R2HG perturbs, usually by inhibiting various and sundry enzymes that would usually use the cofactor 2-oxyglutarate. So a number of companies, including Agios, were developing inhibitors of these mutant enzymes. But the review articles began to suggest that maybe the effects of 2HG would not be reversible because a lot of the targets of 2HG are involved in epigenetic reprogramming. And so the naysayers started lining up saying this will never be reversible. And so to begin to address this, Julie Lossman in the lab obtained a tool compound from Agios that prevents the production of 2HG by mutant IDH. And fortunately, she had already created a cell-based system where mutant IDH would score as an oncoprotein. And in particular, she generated modified versions of a leukemic cell line called TF1 that were now wild type for IDH1 or had this canonical IDH1 mutation. She first measured 2HG levels. And so as expected in the IDH mutant tumor cell line, this leukemic cell line, there are now millimolar amounts of 2HG. The Agios inhibitor lowers by about 10x, but I should point out doesn't normalize the 2HG. So at least it's having its intended pharmacodynamic effect. And Julie had shown previously that if you take these cells and deprive them of cytokines, the wild-type IDH1 cell will stop proliferating, shown in red. In dark blue are the cells with the IDH1 mutation and now treated with the vehicle. And now she's ready to do the experiment. She can take these cells, treat them with the Agios inhibitor, and as she had expected, or hoped, I should say, she saw impaired proliferation when she took the IDH mutant cells and treated them with the inhibitor that's going to block 2HG production. But again, I always tell the students the most dangerous result in science is the one you were hoping for, because that's when you declare victory and you get lazy. So the rosiest interpretation, again, would be, yep, everything's on target. The model is correct. 2HG is responsible for this increase in proliferation. And if you prevent the 2HG from being produced, the cells stop proliferating. The least interesting interpretation, however, is that this drug does prevent 2HG production, but it's also a poison. And because it's a poison, these cells aren't very happy, and because they're not very happy, they're making a little less 2HG, and because it's a poison and they're not happy, they're growing not very well. Is that really our final figure panel? Is that really figure 6F? Do we declare victory? But Julie's much more careful, and she realized if this is really on target, if I do this experiment, but instead of giving them mutant IDH, I give the cells an esterified version of 2HG that enters the cells and then gets trapped at tumor-relevant concentrations, those cells should be completely insensitive to the drug, and that's what she saw. So if you give the cells 2HG itself in a version that's now cell membrane permeable, these cells don't care about a drug that blocks 2HG production. And not shown here is if you give this ester to these cells, now they grow like that. So that's on target. Also, unfortunately, these drugs did go into the clinic for the treatment of IDH mutant leukemia for technical reasons. The inhibitors of mutant IDH2 went into the clinic before the IDH1 inhibitor. This happens to be early data from the phase 1 trial with the IDH2 inhibitors. But suffice it to say that some patients had dramatic responses. In fact, some patients went on to complete remissions and had consolidated transplants. But now that the dust has settled, only about 30% of the patients have these sort of remarkable responses. So again, I think we just have to acknowledge that heterogeneity is the rule, not the exception. So one of the last examples of hopefully logic and rescues and necessity and sufficiency, et cetera, is the work that our lab did and Ben Ebert's lab did on thalidomide, which I think many of you may know was an infamous molecule that was available over the counter in the middle of the 20th century, largely in Europe, less so in the United States, thanks to the FDA. But this drug was available over the counter for morning sickness and insomnia, and unfortunately some women who took this drug during pregnancy gave rise to children with these horrific limb defects, and they came to be known as the thalidomide babies. Then, through sheer serendipity, thalidomide and its cousins, lenalidomide and pomalidomide, were discovered to be almost wonder drugs for certain B-cell malignancies, and in particular multiple myeloma. And so then the question was, how does this drug even work? And so the clue came initially from Hiroshi Handa's lab, where he immobilized thalidomide on a cephros bead and looked for thalidomide binding proteins, and he identified a ubiquitin ligase that contains cereblon as the substrate recognition subunit, cereblon performing a function analogous to VHL in its ubiquitin ligase complex. And then, perhaps because reviewer 3 demanded it, the last figure of this paper showed that if you add enough thalidomide in a test tube with the cereblon ubiquitin ligase, you can block cereblon's auto-ubiquitination activity. And so in 2010, it looked like the IMiDs were simply cereblon antagonists. But it's been said that science is organized common sense where many a beautiful theory has been destroyed by an ugly fact. So the ugly facts here were, if you looked at patients with multiple myeloma who had initially responded to an IMiD and then became resistant, and you looked at their resistant myeloma cells, not infrequently, the resistant myeloma cells were no longer making cereblon. Conversely, the higher the levels of cereblon, the more likely you are to respond to an IMiD. Now, there are lots of explanations for this, but it's hard to reconcile this observation with the thought that you're killing myeloma cells by depriving them of cereblon. Rather, getting rid of cereblon somehow makes you resistant to the drug. And it was sort of interesting to reread the discussions in these earlier papers, because they all said things like, this proves for the first time that cereblon is the target of the IMiDs in myeloma. But no one was willing to stick their neck out and say, and it ain't simply an antagonist. It has to be a neomorph. But Ben's lab and our lab simultaneously thought this has to be a neomorph. Cereblon, once bound to an IMiD, is doing something that unbound cereblon can't do. We imagined that the drug was repurposing cereblon to destroy a myeloma-relevant oncoprotein, and so Gang Lu in the lab made a vector where your favorite protein of interest is fused to firefly luciferase. And he then encoded on the same transcript Ranilla luciferase, both under iris control. And now he deduced that he could use the ratio of firefly to Ranilla to infer things about the changes in the stability of the fusion partner. And so he actually created a library of about 12,000 of these and arrayed them in 96 well plates. Each well has a different fusion protein, and he then, with the help of our colleagues at the Belfer Center, either treated the plates with lenalidomide or vehicle, and he looked for examples where lenalidomide stabilized or destabilized the fusion protein as determined by a change in the ratio of firefly to Ranilla. And since this is all published, I'll just tell you that the two true positives in this screen were IKZF1 and IKZF3, which are two transcription factors that play very important roles in lymphocyte biology. So we have plausibility on our side. And note, they're downregulated by the drug, and we went on to show that this was actually cereblon dependent. Gang did secondary assays that actually showed this was quite specific. So for example, here's the validation in low throughput mode of IKZF1 and IKZF3. In red is the ratio of firefly to Ranilla in the lenalidomide treated cells versus the blue is DMSO. If you replace the luciferase tag with an HA tag, you still see downregulation. And here are three closely related transcription factors, and you can see they're completely insensitive to the drug. Now, our biochemical studies made the prediction that maybe the drug was binding to the cereblon substrate adapter unit like a piece of gum. And so we were very happy to see the work of Eric Fisher and Nicholas Thoma, where, in fact, that prediction turned out to be correct. And in a published work, it does appear that that's largely what the drug is doing. It's acting like a piece of gum. I hope the structural biologists won't be offended when I use that metaphor, but I'm not a structural biologist. Okay, but now it's plausible IKZF1 and IKZF3 were the targets, and there's a correlation, of course. They're downregulated by the drug. Are they really playing a causal role? So the first thing that Gang did was he made 3 different hairpins against IKZF1 and 3 different hairpins against IKZF3. And for reasons we don't completely understand, when you knock down IKZF1, you knock down IKZF3. When you knock down IKZF3, you knock down IKZF1. And you also knock down various downstream target genes, such as IRF4. So then what he did was he introduced these hairpins into myeloma cells with one color being present, such as GFP, and he mixed them with cells that had a second color and a control hairpin, and then just did competition assays asking, do the cells where there's knockdown of IKZF1 get selected against over time? And the answer is they did. So, so these cells are suffering a loss of fitness consistent with IKZF1 loss potentially causing an anti-myeloma effect. But again, I hope the theme emerged today, down assays versus up assays. I would still argue this is a down assay. Is it really that interesting that when you knock down some important transcription factor, the cells pay a fitness penalty? Is this really a good target? And so, again, we have correlation on our side, we have plausibility on our side, and now we have a down assay on our side. Can we do a little bit better? So Gang realized the way to do better was to now make an IKZF1 or an IKZF3 that is insensitive to thalidomide. And the way to do that was to recognize there were only 7 amino acid differences between IKZF2 and these 2 guys over here. He then changed each one at a time until he found the critical residue. And now he could do an up assay or a rescue experiment where if the cells have the drug-resistant IKZF1 or the drug-resistant IKZF3, now these cells are largely insensitive to an image, compared to, for example, trying to force the expression of one of the downstream targets, IRF4. So we think this is on target as well. So what kind of things keep me up at night? I'm quite sure that some of the assays we all measure are a combination of on target and off target. And depending on whether that's a Boolean and or a Boolean or, the rescues will or will not work if you go through the logic. So if you really needed both of these to get your phenotype, then the rescue experiment will work, but you'll still be blind to the contribution of the off target. The other thing that keeps me up at night is it's quite clear to me from many discussions I've had that there's another hidden bias in these experiments, which is you don't get to be a negative control very long if you have some unexpected effects on cellular proliferation or viability. So in other words, we're pre-scrubbing the negative control for any badness. And so I've heard more than once people say, oh, you want to use this as your negative control because I can guarantee you it does nothing. And then you compare that to the thing you just bought from Dharmacon or who knows where. And so there's a bias because already the negative control has been pre-selected to not do anything negative to the cell. I also, to my horror, learned in even one of my own lab meetings that most of the young people don't remember the day when, if you were doing an experiment with a peptide or an oligonucleotide, an essential control was to take that sequence and to randomize it or to scramble it as a control for things that might be related to, for example, the physicochemical properties of the peptide or the oligonucleotide. So that was your scramble control. But now we've learned that scramble means that random negative control you got from your buddy's freezer down the hallway because they promised it wouldn't do anything negative in your assays. Lastly, I think there's some sociology at work here, and this is where I'm going to probably end. When I was younger, papers were maybe one major thing being claimed, but you took great pride in how many corroborating lines of evidence you could come up with to support your claims. And the scientists I admire usually had non-obvious experimental approaches to marry to the obvious experimental approaches. But I think, again, in the quest to be more impactful and more translatable, now papers are multiple things, each proven one way, because everyone's racing to get to that final figure that's going to be seemingly somewhat translational. And so I wrote this little piece this last year. And then as a tribute to my wife, I wrote this piece, which summarizes a lot of things we talked about today, plus some other things we didn't have time to talk about today. And I think it's to say, I think collectively we all have to do a better job at this. And if it feels like I'm on a soapbox here, some of you know my wife, who was a leading breast cancer surgeon in Boston self-diagnosed herself with breast cancer in 2003 and survived, but then in 2010 developed a glioblastoma. And thankfully, with modern therapy, we were able to keep her alive for 4.5 years, which wasn't long enough, but it did mean, for example, her seeing her eldest child graduate from college. But one of the many frustrations treating my wife was that everything I had available to treat her was really based on science that was done 10 or 20 years ago. And God forbid, one of my children or grandchildren gets this disease, and frankly, statistically, this disease is going to touch every one of you at some point. They're going to be relying on the science we're doing now. So I think we have to get away from what Chuck Shear calls CNS disease, which stands for cell, nature, and science. Those are the false gods. Of course, we love to publish in those journals too. But the first question should be, is this true, robust? Is someone likely to be able to build on this? Or am I simply doing this to pad my CV? And if it's the latter, please stop, because I can tell you you're becoming a dominant negative, and I have no more tolerance for the dominant negatives. The last thing I'll say about my wife is she never gave up. And to the young people in the room, please don't give up, again, in part because I'm thinking about my children and my grandchildren. There's no shortage of naysayerism in clinical oncology, and I'm sure you're aware that many people are talking about the fact that responses to targeted agents are often short-lived as monotherapy, and that's certainly true. This, I think, is the most famous image currently in targeted therapy, so I don't even probably have to tell you. This is a patient with metastatic melanoma that had a BRAF mutation. They were treated with the Plexicon BRAF inhibitor. They went into this Lazarus-like remission, and then months later, they're right back in hot water. And so both in the lay press and the scientific press, you can see that some people are getting a little nervous. Is this the right road, or is this not the right road? And I can tell you some people who have some clout, at least in some circles, think we are on the wrong road. So if you have heard Jim Watson speak, or you read this paper, he wrote that given the seemingly almost intrinsic genetic instability of many late-stage cancers, we should not be surprised when key old-timers, and I'm not sure if I'm now a key old-timer, in cancer genetics doubt being able to truly cure most victims of widespread metastatic cancer. And the first time I saw him, I said, way to rally the troops, Jim. If I was a 22-year-old thinking about a career in cancer pharmacology and cancer biology, that would really get my juices going. But of course, he's forgetting something, and I think many of us have forgotten things they used to teach in the 70s, and they still taught in the 80s, but I'm not sure they teach anymore. And that is, the old-fashioned blocking and tackling of combination chemotherapy. So let's go back to that patient who had those subcutaneous nodules. So even a tumor of a centimeter cube has about 10 to the 9 cells. The tumor burden in most cancer patients is 10 to the 10 to 10 to the 12th. So even if you had a great drug like the Plexicon drug, but 1 in a million cells were resistant, maybe on an epigenetic basis, your chance of cure with a single agent is approximately 0. And you get the picture I just showed you a moment ago if you do the back of the envelope calculations. But fortunately, there's a playbook here. It's the playbook that's worked before, and that is to make the math work for you. And the way you make the math work for you is you combine drugs that have distinct mechanisms of action, and because they have distinct mechanisms of action, they're not cross-resistant with one another, and they have toxicities that don't overlap in a prohibitive way. That's the playbook. We know the playbook works because it's been done multiple times before. This is what the treatment of tuberculosis looked like about 100 years ago in the United States before the advent of combination chemotherapy for TB. When I was a resident at Hopkins in the 80s, there was a time when some of the AIDS wards were starting to feel a little bit like this. But combination chemotherapy, I think, will set you free. Lastly, because the words that come out of Washington and the words that come out of Bethesda really, really matter. When I was on the BSA, I remember talking to Rick Klausner once, and I said, Rick, it must be really hard to go to the Hill and talk to Congress, because they must occasionally say things like, when Kennedy said we're going to put a man on the moon in a decade, we did it. Nixon started the war on cancer in 1972, and you guys are still stuck doing trench warfare. When is this going to be over? When are you going to get your act together? And what Rick said was absolutely correct. Putting a man on the moon was a spectacular engineering feat. It was not a scientific feat. Galileo and Newton had already come and gone. The basic scientific principles you needed to know to put a man on the moon were known. At that point, it had been reduced to engineering. Unfortunately, cancer is still quite a bit of science to be done. I think there's some that's been reduced to engineering. But if you don't understand the sociology and the differences between science and engineering, you get policy completely wrong. So at the risk of oversimplifying, science is learning the rules, engineering is using the rules, and I'm going to plug a second book called The Idea Factory, which is the history of the Bell Labs. They understood this. Okay, and mind you, the people who do science well are often not good engineers and vice versa. In fact, science is often dominated by individuals, sometimes referred to as the lone hunter-gatherer scientist. Engineering is usually accomplished in teams. Because of the resources, engineering is usually top-down. Really disruptive science is often profits from bottom-up. And in particular with science, I mean true science, the timelines are unpredictable. You're really limited by knowledge. And frankly, if you can predict the deliverables in 5 years, you're probably not really doing science. Whereas in engineering, you can ask for timelines like that decade. You can approximate how much it's going to cost, as Kennedy and his administration did. And the deliverable in this case was putting a man on the moon. So I would argue, I think the moonshot in cancer is well intended, but it's absolutely the wrong metaphor. And again, just to give you one more example of the sociology, because this is written about in the lay press all the time. If you compare science and engineering, if you're going to put a man on the moon, you want everyone going in the same direction. But people who study how truly transformative science gets done would tell you, especially in the early days, you'd be better off having 10 people going in different directions, looking under rocks, and hoping that one or two of them actually discover something. If you make all your scientists do all this, you're just creating a herd mentality, which I think is an absolute disaster. And so I would summarize by saying every good example of translation I can think of really happened the same way. There was a line of investigation that matured to the point where you knew enough to know what to do. It didn't happen because you put a gun to some poor young person's head and said, go do translational research. And so just as an example, suppose this was the identification of the BCR-ABL translocation in CML. A lot of legwork had to be doing before, Alex Matter at Novartis and Charles Sawyers and Brian Drucker said, if you only had a drug that inhibited BCR-ABL, maybe you could go all the way to the clinic and make an impact on CML. So I like to point out that historically the secret sauce in biomedical research in this country was to acknowledge that this is how it happens, to acknowledge that our friends out here, which we could also equate, I think, with the biotech and pharmaceutical industry, they're actually pretty good at this. They can't do this because they can't justify this to their investors. And so we had this beautiful symbiotic relationship going that they rely on, which is they will do this and we will do this. And it even goes further, of course, because you can even replace some of this earlier stuff over here with the stuff that made it possible. So when I go to the Lillys and the Pharmas of the world, they tell me, if you guys want to become more translational, that's great, but we can't do science. We're pretty good at the engineering part. If you tell us this is a really good target, we can make a drug. But please don't stop doing the stuff on the left because we can't do the stuff on the left. And so that's where I will stop and just remind you that this seems to come up about I'm clocking it now, about every 10 years there's an article that comes out that doesn't get this distinction and says, if you would only get your acts together, if you'd only work in teams, if you only had proper business plans, you would be done. Okay, so please remember it's a combination of science and engineering. Thank you, and thank you for your indulgence.
Thank you very much, Bill. We do have time for one or two questions. There's microphones in each of the two aisles.
Or maybe there are no questions.
Just a comment. There was a paper in Nature or Science, I forget which one, where they said they should allow all scientists only a certain amount of words in their lifetime, in their scientific lifetime. And if you did that and if you allotted that, you would increase the overall quality of the literature.
Let me push back on that a little bit. I think there are other things that have contributed to this mess. So, for example, a number of people, even before I speculated it, speculated that Ben Lewin's simple act of putting the methods at the back end of the cell papers began to de-emphasize the importance of how the experiments were actually done. And so I worry that by creating artificial word limits, we're actually going to run into this problem where I read many papers and frankly I almost can't review them because I don't understand the experiments in sufficient detail to even be critical. And so I like the sentiment, but I think we have to do much better jobs of explaining to one another how we did the experiments, and I think it's the other price we pay when we try to combine multiple papers into one paper, and then worse yet, move to a letter format when really there's a lot of sophisticated, nuanced work that's going on that has to be understood in greater detail. So I think it's one for debate, but I think now maybe a part of the solution is to put more information in the supplements. in terms of having online protocols in sufficient detail that people can replicate the results. But I think it's an interesting point to discuss.
Thank you.
Well, that was a really, really inspiring talk, so thank you for that. What I wanted to ask was, so this is wonderful, everything you're proposing. It's how we all come into science and want to originally do our science, and then we all get bogged down by there's so much competition, you need a CNS paper if you're going to succeed, and then even if you're on tenure track, you've got a limited amount of time to even perform and get your papers out, etc. So in order to achieve this in the real world and to stop us making mansions of straw, shouldn't there be a top-down reorganization of how we pull our resources? So people who are giving the tenure tracks the money should say, okay, you have a bit longer, and yes, it would be great if you have a CNS paper, but we also are going to look at some lower papers, and we're also going to look at how many times they're cited and how far and how much this work has expanded into other things. And that's what we're going to judge you on. Yeah,
it's a great question on multiple levels, so let me toss out a couple ideas. So the first step in dealing with any problem is acknowledging there's a problem. I think that's happening now. There is a problem. The second is, and this hints at your question, we collectively helped make the problem, so I think we collectively start to undo the problem. And some things will require some collective effort to maybe change the way we review papers. It could be as simple as I heard, not since I'm now an old-timer, I have to be careful to use the phrase old-timers, but people, even one generation beyond me, tell me that, when they used to review a paper, they would review a paper with the default position being, this should be accepted unless it's clearly flawed, because everything else being equal, it would be better to get this out and allow people to read it. I know many people now, I think, start with reviewing papers with the default being, I'm going to find a reason to reject this, or this will be rejected unless it's perfect. And of course, trying to reach perfection is an asymptotic function. You never quite get there. So I think we have to really step back a moment and understand it takes more courage and more experience to accept a paper than to reject a paper. I think we should go back to what is the principal conclusion of this paper, and is this conclusion likely to be true in 10 years based on the data in this paper? Okay, so I think we have to change the way we review papers. I really think we have to change the peer review system a little bit. I think even putting in things like requests for timelines and deliverables and impact statements has unintended consequences in terms of the kinds of questions people pursue, how they pursue them. Now, I'm old enough and I'm well-funded enough that if I see something really exciting, I don't feel wedded to keep going down the road I said I was going to go down. But I can imagine a lot of young people feel like they have to go down that road even though there's a great opportunity that just arose, but it wasn't in their year 3 timeline. So I think we have to go back to acknowledging we're doing science, and think of how many magnificent scientific discoveries you couldn't have predicted the impact a priori. It was only in hindsight, CRISPR being yet one recent example, but I can think of many other examples. So I think you have to have reviewers with scientific taste and have a nose for where there might be a discovery to be made, but not have every grant start out with, there are 100,000 patients each year with lung cancer and there's a pressing need for new treatments for lung cancer. If I read that one more time, how about I have this really interesting puzzle I've stumbled onto, and I just think it might lead to new insights into some interesting area of biology. Now, the nice thing about cancer is it's not that hard to imagine how almost any biological phenomenon could loop back to cancer. And so, I think we just need to be a little more Catholic as to what cancer research is and not have to have everything be viewed as being, 5 yards from a touchdown. I think it's okay to have some grants that are back there on the 50-yard line,
Thank you.
It's not so much a question, but I really did appreciate towards the end of your talk in providing sort of a meta view in terms of the symbiosis that exists between science and engineering. There are 2 quotes and 2 metaphors that have also stood out just in reading. One is that part of engineering is making tools, and I don't know who said it, but someone said that first we make our tools and eventually our tools also make us.
Yes.
And then the other point or metaphor that I've kind of come across, which I thought was really illuminating, is the idea of a cathedral and a bazaar. In a science, it is kind of a bazaar. In engineering, you have a cathedral. Both approaches are necessary. And oftentimes the bazaar comes up with new plans to deconstruct the cathedral and rebuild it again.
Yes.
I think all of human history, as we go through from essentially the Renaissance till now, we have been deconstructing and constructing cathedrals through the ideas that we get from the bazaar.
Yeah. And I think those are good points. And I don't mean to disparage one or the other. They're just different, types. Clearly, I love my iPhone. My iPhone was made by engineers, But, I think there's real power and importance to both groups. But I think you have to acknowledge that you need both. And then you have to look in the mirror and say, who are you? For example, the other analogy I point out is, I think I sometimes maybe unfairly say, engineers live for expected results. I think a good scientist lives for the unexpected result that changes the way they thought about something. So I don't think the NASA engineers would have, been going high-fiving each other if they'd missed the moon because of something they hadn't anticipated before. I mean, I think they wanted to get to the moon. But how many scientific discoveries really started with a completely unexpected result that just changed the way you think? So I actually worry if all your results are the expected results, I think you're starting to gravitate towards engineering. I hope occasionally you're getting really, really surprised, and I hope sometimes those surprises are the first step towards a meaningful discovery. Okay, great, thank you.
Thank you very much.